Alright guys, in the last video we talked about the chromosomes in the nucleus. If we're going to talk about cell division, we really do need to discuss the contents of the nucleus and kind of what happens to the nucleus during different parts of the cell's life. Okay, and today we're going to discuss more detail about each one of the phases of the cell cycle. Now, one of the things I don't know if I made abundantly clear, but when we talk about this term, the cell cycle, I actually am talking about the entire life cycle of the cell. Not necessarily from life to death, but more from not necessarily from birth to death, but more from birth to next cell division, really is kind of what we're talking about when we talk about the cell cycle. The cell's life is oftentimes characterized in that manner when we discuss the cell cycle. So that's what we're going to get into here today, and we'll discuss the phases of the cell cycle. It consists of 2 to 3 really big phases, and then we can break those down into much smaller phases. The main phase of the cell's life cycle is interphase. Okay, interphase makes up about 90% of the cell's life during a given day. So if you could break up the 24-hour period into chunks, 90% of that 24-hour period is going to be spent in interphase, and the rest of that time, about 10%, is going to be spent dividing. Okay, so interphase is where the cell grows. That's where it copies its chromosomes, copies its DNA, and then prepares for cell division. Okay, so that is something that can then also be broken up into subphases. If we take all those things, the growth, the copy of chromosomes, and then preparation for cell division, we take all that stuff into account, we can then split that into other phases, and they are the G1 phase, the S phase, and the G2 phase. Okay, now the G1 phase officially, I guess, stands for first gap. I probably should know why they call it the first gap phase, but I don't, and it's really not a big deal that you do either. But I like to kind of alter it a little bit and make it so that it's easier to remember what actually happens in the G phase, or in the G phases. In the G phases, we have a lot of growth. Okay, so from the time the cell is quote-unquote born, it is actually relatively small because we previously took a big cell and split it into 2, basically chopped all the stuff that it had in half, right? And so in the G1 phase, that part of the cell's life is actually spent growing. All right. The S phase, is where we have the synthesis of DNA. Okay, that's where we get the term S phase. Okay, S stands for synthesis. Now, what does it mean to synthesize DNA? It means to make new DNA. So in the S phase, we are actually spending time replicating our chromosomes inside of our cells. Okay, that's the main thing that happens in the S phase. The G2 phase, yet we have another growth phase. Okay, now in this growth phase, the cell does spend a little bit of time growing. It makes some extra cytoplasm, maybe a few extra organelles, and it prepares itself for cell division. So in this G2 phase, we have a preparation of cell division. Okay, so it is a point at which we are then, after the G2 phase, we're going to start the mitotic phase. Okay, this is where the cell spends about the other 10% of its life. Okay, and we can put that mitotic phase kind of in this nice pie chart that shows the relative amounts of time that the cell actually spends in its life, in each phase in its life. So in the big bluish green arrow that we see out here, we have all of interphase, which we've just talked about can be broken up into the G1 phase, the first growth phase, the S phase, where DNA is replicated, and then the G2 phase, where we start to prepare for cell division. And the reason we prepare for cell division is because that's what's next. So the mitotic phase is where we get division of the nucleus, which is mitosis, and then where we get the division of the cytoplasm, which is cytokinesis. So mitosis and cytokinesis together make up what we call the M phase, or the mitotic phase. So the cell's life cycle can be broken up into 2 big phases, interphase and then the mitotic phase. And each one of those, interphase and the mitotic phase, can be broken up into much smaller phases, okay, as we see here in this chart. So when we work our way through this stuff, we're not going to talk much about specifics in terms of what happens with each of these things in interphase. You know, there's a lot that happens here. You know, we'd spend all day trying to figure out all the different things that happen here, and that's what scientists are trying to, you know, or scientists do. They know what that is, but there's just too much to talk about and worry about in terms of learning. Now, with the mitotic phase, we can spend time and discuss some of the details of those phases of mitosis and cytokinesis, which is what we're going to do here today. So these should be a little bit of review. You know, I know that a lot of teachers, when you're in 6th, 7th grade, start this stuff, right? They talk about, okay, cells divide. What happens when cells divide? This is the phases they go through. And, you know, we're going to do something of that as well. You're going to have to know some of the big events that occur, but I'm not going to have you go crazy in the details. So when we talk about each one of these phases of mitosis that you see in front of you, I'm going to talk about some detail, but just not crazy detail. And so we'll kind of speed right through it. So you may want to have the notes ready and jotting down exactly what I talk about in those particular phases, because I'll go pretty quick, okay? Now you can always rewind the video, I suppose, as well. All right, so mitosis can be divided up into, some people say 5, I sometimes say 4. It all depends on your opinions of pro-metaphase. In my opinion, pro-metaphase is kind of like this hipster phase, and somebody wanted to like invent something, so they made up this phase. I don't know. I've always known it as PMAT, right? Prophase, metaphase, anaphase, and telophase. And if you want to incorporate pro-metaphase, you're more than welcome to do so. But you'd have to grow a really long beard and get some mustache wax and you know, things like that. So cytokinesis is the final stage of the mitotic phase, and we'll talk about that a little bit as well. Okay, so let's get into some of the details. And like I said, I'm going to go quick here, and I've got a lot of these phases are all just together, okay, in one big picture. And I'm going to talk about each one of these in general, more generally, add a few details when needed. Okay, so we see all the way to the left here. They've depicted G2 of interphase, and so there are some important things that happen in G2 of interphase. So at the end of G2, we're starting to round up and be done with interphase, and we're going to start to transition into the mitotic phases. And we notice here that in G2 of interphase, we've got a massive amount of genetic material in here. Okay, we've got a lot of chromatin because our DNA has been duplicated. And so we've got double the amount of genetic material that we normally do because we've already gone through synthesis. Okay, so that's the big thing I want you to take away here. And we also know that we've started to create these thing called centrosomes. And I'm going to spend a lot more time on centrosomes and microtubules after I'm done with all the phases here of mitosis. Okay. So after G2, we get a transition into the mitotic phase, into mitosis, and we go right into prophase, which is what we see in the second picture, the middle picture. And we see some different things happening here. We see some definite changes between what we saw in interphase and what we see now in the mitotic phase. We start to see, number one, the centrosomes have these little mitotic spindles, this is what we call spindle fibers, kind of reaching out, being built across the cell. We notice that the chromosomes have condensed. Okay, here they look like thin spaghetti. Here they look like little butterflies. Okay, so they're starting to condense here in prophase. And we notice that each one of these has a sister chromatid attached to another one. We'll discuss a little bit more about that. Okay, as we transition into the hipster prometaphase, the hipster prometaphase is going to have a little bit of change here compared to prophase. Okay, a lot of people just lump these both together as prophase. But at the end of prophase, or at the end of prometaphase, we start to see that the chromosomes are now fully condensed. They do look like these butterfly structures, whereas in the previous picture they kind of look like they're still starting to condense. And we notice that the centrosomes have migrated to either pole of the cell. Okay, we see them at the very ends here, look like rigatoni noodles. And we see that the nuclear envelope has started to degrade or break down, because we need to make sure that we can move those chromosomes during division. We can't have the nuclear membrane kind of holding them in the same spot as they started. So we have to break down the nuclear envelope so we can move those guys. All right. So after prophase, prometaphase, whatever you want to call it, we do transition into metaphase. Okay. Now, at some point there between prophase and metaphase, the spindle microtubules, okay, the little strings here that we see, those things have attached to the chromosomes, okay, and they've attached to the chromosomes at these little black dots. Those little black dots are called kinetochores. I'll add more to the picture about what kinetochores are in a few minutes. And so these little spindle microtubules have attached themselves to the chromosomes, and they have allowed the chromosomes to line up at what's called the metaphase plate. The metaphase plate is nothing but like this kind of invisible line that goes right down the middle of the cell. And it's a place where the chromosomes will line up during metaphase, okay? So once this happens, once they've kind of been lined up, and this is it's actually kind of an important step for the cell to organize the chromosomes this way, so that they can make sure that that they get separated, the sister chromatids get separated appropriately, and so they want to be lined up top to bottom. Okay, it's kind of important that you notice that these guys are all lined up top to bottom. Okay, because that will change in meiosis, which is the other kind of derivation of this process. All right. Next, we transition into anaphase. In anaphase, we notice that something has occurred that is relatively important. We notice that the sister chromatids that we see were attached in the previous picture have now separated from one another. Okay, so these sister chromatids then start to travel back on the microtubules towards either end of the cell, towards either pole of the cell. And so you just notice that this one here was ripped away from this one here, and they travel to opposite ends of the elongating cell. And that same thing happens to all of the other chromosomes as well, or sister chromatids. All right, the final stages of mitosis include telophase and cytokinesis. So telophase sees the ending of movement of the chromosomes. It sees the uncondensing of the chromosomes. We see that they start to turn more like into spaghetti strings, and they're no longer condensed like they were in the previous picture. And we start to see the reformation of the nuclear envelope, okay, the nuclear membrane. So all of these things mark a point where we see that the cell is starting to return back to normal, okay? And cytokinesis is a term that means division of the cytoplasm. And that's what we see happening right down the middle here. Something called a cleavage furrow is created, and that happens in animal cells. And in plant cells, instead of a cleavage furrow, we see something called a cell plate formed. Okay, and that cell plate formed is something that basically allows the cell wall of the plant cells to be created as the two cells that have just replicated start to differentiate from one another, or separate themselves from one another, I should say. Okay, so telophase is basically just wrapping everything up, getting everything returned back to normal. And one thing I do want you to note, and because it'll play a role in the cell and kind of our discussion of the cell, take note of the size of these two daughter cells. Okay, they are effectively half the size of the previous cell here. Okay, and that comes into this idea of the cell after it's been created. It has to go through these growth phases and these preparation phases before it can divide again. You just wouldn't have enough stuff in this little cell right here to divide rapidly again. You have to make it grow and add some of the organelles that it might be missing and things of that nature. So those are your phases of mitosis. Like I said, I kind of sped through them, but it's something that you don't have to spend a ton of time on, and I'm hoping that it can be kind of basic review, since a lot of you guys probably have seen this in some capacity already. Okay. I do have a computer animation of this. I think I'd rather show that in class when we actually are here. So I think I'll do that in class. It's actually quite... Is it lengthy? We're at 14 minutes. Let's just do it. We'll do it right now. Okay. And no, yeah, let's allow it. We'll do it right now.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right, so there you have it. I like it. It throws everything into perspective. It does it relatively pretty quickly, and it's something now you can go back and check it out and see, if you need a review on it, it's there for you, okay? So I do want to spend a little bit more time here on, this is maybe a pet lesson for me, because it addresses something that I was never taught, and when I learned about it, I was like, oh my gosh, that's mind-blowing, it's crazy. But yeah, I'd like to take a little bit closer look at the mitotic spindle, and it's probably one of the more, again, I like to spend a little bit of time on some of the things that don't get all the publicity and the attention, and that would be this thing called the mitotic spindle. The mitotic spindle is a bunch of microtubules that control chromosome movement during mitosis, and we saw them in the diagrams. They started at the little rigatoni noodles that I had mentioned, and they grew from there, and they look like little spaghetti strands that had grown out of these things. And so the mitotic spindle is actually composed of many things. It's composed of the spindle microtubules, which look like the spaghetti, the asters, which look like almost like little starbursts that come out of this thing. Now you'll see pictures of them here in a second again. And then the centrosomes, which I said look like those kind of short, fat noodles at almost right angles to one another. And so each one of these things, these components, together make up the mitotic spindle. And the mitotic spindle is, as I mentioned before, responsible for moving the chromosomes from place to place. So to start this whole entire process, the cell creates these things called the centrosome. Okay? The centrosomes will replicate, or the centrosome will replicate, and each one of these little centrosomes will migrate to either end of the cell that is elongating and dividing. And the spindle microtubules, I should have highlighted those in yellow right here. The spindle microtubules start at the centrosome, and they radiate outwards. They extend outwards towards the chromosomes. And when they reach the chromosomes, they will attach to something called the kinetochore that is on the chromosome. And so we'll see all of this stuff in the pictures coming up here, okay? And final bullet point there, something known as the aster radiates out of these centrosomes and basically act as an anchor to keep the entire complex in one spot once it gets to where it's supposed to go, okay? So look up at this picture at the top here. We see the centrosome. It looks like these two short noodle complexes that are at right angles, okay, 90-degree angles of one another. That's just how they look. I'm not really entirely certain why they are at 90-degree angles, but they are. We see this thing called the aster at the top here, which radiates out from the centrosome, okay, and the aster acts almost like it digs in, almost like claws that dig in. It kind of looks like claws too, right? They dig in to a certain part of the cell, probably components of the cytoskeleton is what it anchors itself onto. And then we see that the mitotic spindles, or sometimes called the spindle fibers, radiate out from the centrosomes, okay? And so these mitotic spindles are going to radiate out to the chromosomes. They're going to attach themselves to the chromosomes. And if you look really closely on this diagram, there's little black dots in the center of each of those chromosomes, right? And those little black dots are called kinetochores. All right, kinetochores are what the microtubules attach to. Okay, so they are protein complexes that will attach to the microtubules. Now, some of these spindle microtubules, if you notice, will attach to the chromosomes, right? They'll attach to the kinetochores. We call these kinetochore microtubules, all right? We have others that do not. Kind of you see some of those right in the middle, in between the chromosomes. We have other spindle microtubules that do not attach to the kinetochores, and obviously those would be called non-kinetochore microtubules, okay? Now, even the non-kinetochore microtubules have a job, okay? And then we'll get to what that job is in a second. But what I'd like to talk about here is kind of my misconception when first studying this stuff, when I first started learning it. When you see these diagrams of the chromosomes moving back towards the poles of the cell, it almost looks as if these little centrosomes that we see up here, looks like these little centrosomes that we see up here are almost like fishing rod and reel, right? I'm going to throw out a fishing line, grab myself a fish, and then reel it back. When we see that happen in the diagrams, that's kind of what it looks like. Let's go back to the diagram here. It looks like, okay, we've got the fishing line attached to the fish, and how do we get it back? Well, the centrosome reels it back. That's what I thought for 26 years of my life until I started teaching this class. I paid $40,000 in tuition and room and board at U of I, and another 18 at NIU, and I feel like I got cheated out of my money. I never learned what I'm about to tell you here, but that's not actually how it happens, okay? So as these chromosomes start to move back towards the poles, don't think of it as the centrosome pulling them back like a fishing wire. Think of it as the kinetochore, think of it as the chromosome hitching a ride on the microtubule highway. Remember we mentioned when we talked about cytoskeleton, which microtubules are cytoskeletal structures, the cytoskeleton acts as a highway, okay? And the organelles and other things inside of the cell, including chromosomes, can actually ride on these highways, okay? And that's exactly what's happening with these chromosomes. If we take a really, really close look at the chromosome down here, we can zoom in on that kinetochore, which I've talked about a couple times in the last couple of slides. The kinetochore is that protein complex at the center of the chromosome. And if you look even closer, it has something called a motor protein, okay? Motor proteins are like little trucks, like U-Haul trucks, that kind of move back and forth on this microtubule highway. And so what we see inside this kinetochore is it has a couple of these motor proteins embedded inside of them. And so what these kinetochores allow the chromosome to do is hitch a ride. This kinetochore acts like a U-Haul truck, and this giant chromosome is hanging off the back end of it. Okay, almost like a big RV kind of, right? And so we've got this kinetochore, which acts as like a U-Haul truck, and then the chromosome, which acts like a giant pull-behind RV trailer. And the motor proteins drive back this way. Okay, if you follow the cursor, drive back this way with the chromosome attached. And then so what happens is that after that road is used, okay, it's not needed anymore, so the cell breaks it apart. And that's what you see here where it says tubulin subunits. That's just evidence that says that the road is broken down after the kinetochore moves past it. Okay? And so they did an experiment to verify this, and I had never learned about it until I started teaching this class. I just thought it was fascinating once I latched on it. I'm like, oh my gosh, it's not a fishing rod and reel. It's actually the chromosome hitching a ride on some U-Haul trucks called motor proteins that are driving on the microtubule highway. Okay, so that's what happens during anaphase. Hopefully I've at least changed your perspective on that one thing, and maybe you learn that and it always sticks with you. Maybe it's the one thing you take away from AP Biology that you'll always remember. All right. Continue on the mitotic spindle here. I mentioned that even though we have some of those kinetochore microtubules that attach to the kinetochores, okay, the famous ones, okay, these guys right here, because they allow the chromosomes to hitch a ride back to each pole, right? Those are what we would consider to be the important ones. And I always like to give the ones that are marginalized a little bit extra publicity. Let's talk about the non-kinetochore microtubules, the ones that miss the mark, the ones that don't hit a chromosome. They still have a very valuable job, okay? And their job is basically to elongate the cell. And we can see these non-kinetochore microtubules down here in the picture. And what those, I'm gonna take a snapshot of this and then draw on it, if it'll let me. There we go. All right. We see that these microtubules are really good at growing. To get as far as they got, they had to grow at some point, right? And so this one at one point started here, and it grew out this way as well. So they would actually continue that course of progress. They would continue to grow, and when they continue to grow, what they're going to do is they're going to push the cytoplasm this way, and they're also going to push the cytoplasm this way, so that ultimately that cell is going to elongate further this way overall and elongate further this way overall. So that cell is going to basically start here and it's going to stretch. And the reason it stretches is because it's got these things inside of it that are pushing opposite ways of one another. So the non-kinetochore microtubules, the ones that don't attach to the chromosomes, actually still do have a job. Oftentimes those are just kind of, in these diagrams we see, they're just kind of left there and they're kind of ignored. Nobody talks about them. They want to be talked about too, and they do have a job, and it's relatively important because we need that cell to elongate when we start to talk about cytokinesis, okay? Cytokinesis needs room to happen, okay? Cytokinesis, again, for review, is the separation or the splitting of the cytoplasm, okay? And it is really the final thing that occurs before we actually get two distinct daughter cells. Now, this process happens differently in plants and animals, and so I want to talk just briefly about each. In animal cells, this happens with the creation of something known as the cleavage furrow. Okay, here we can see a cell that is in the process of cleaving or being split in half, and we can see kind of an idealized cartoon picture of it right down here. And the way this happens is that there are microfilaments. Again, these are components of the cytoskeleton. So the cytoskeleton we kind of breezed over in the cell parts. Again, pops its head up here and has a pretty important role in cell division. Okay, and so these microfilaments act as almost like the drawstring bags. And I usually ask for a student to throw me one of their drawstring bags because inevitably they have one from PE. But you open up that drawstring bag, and that would be like where this contractile ring of microfilaments happens to be located. And as you pull that string, the opening gets smaller and smaller, and that's exactly what we have happening here. The microfilament is something that will get smaller and smaller. We're contracting it smaller and smaller and smaller, and eventually it pinches off that cytoplasm and the plasma membrane so that we get two distinct daughter cells. Okay. Again, I regret that I don't have one of those, but I would imagine you get the idea. Okay. Now, in plant cells, it happens a little bit differently. There's no real good way to cleave the cell wall with the use of a contractile microfilament. It's just too strong. So what the plant cells actually do is they build a cell wall, and they start with something known as the cell plate. Okay, it's a very rudimentary, basic cell wall, and it happens to form right in between the two daughter cells that have been created here, the two distinct nuclei. And that cell plate will grow towards either end of the existing wall, and it will ultimately fuse with the existing wall. And so we can actually kind of see that. Here's up in this microscope picture here, we can see on the right, here's a nuclei, and on the left, here's another nuclei. And in the middle, we get these little vesicles form, and ultimately those little vesicles will fuse to form the cell plate, and then the cell plate will grow towards the existing wall and ultimately fuse with it to form two distinct daughter cells. And we can see that here as well. This is a better image of it. We can see the cell plate. Actually, we can see all of the phases of cell division here. We can see prophase, prometaphase, if you want to go with that. Metaphase here, these things line up at the center, anaphase. And then here we get the separation, obviously, of the nuclei, but here we start to see the cell plate formed in the middle. And ultimately, this thing will fuse with the existing cell wall on either end, and you get two distinct daughter cells. Okay? So that is... those are, I should say, the phases of cell division in a nutshell. Sped through some of it, but a lot of it should be kind of basic review. So we will get in video 3, we will start to discuss how this process is regulated, and that's where this kind of relates to cell communication and communication within a cell. Okay, so I will talk to you guys then. Have a good one.